I'm JMiller, and I'm going to be talking to you about how biofilm formation will inhibit bacterial wilt disease in tomato plants. So my hypothesis is if I'm able to inoculate soil with Bacillus subtilis, then a biofilm will be produced, which will ultimately inhibit bacterial wilt disease. And so what I'm using is Bacillus subtilis. It's a non-pathogenic gram-positive bacteria. It's also a facultative anaerobe, which grows in oxygen or oxygen-deprived environments. And what the Bacillus subtilis is going to do is actually produce a biofilm around the tomato roots. And this biofilm is it's made by single cells separating into permanent attached cells and is complemented by the formation of a exopolymorphic matrix. And then so what it's supposed to do is actually prevent the Ralstonia solanacarium from entering the roots. And this is a gram-negative bacterium. It has a host range of 200 species and can survive in soil for about 12 months. And it actually creates the infection in the tomato roots by entering small wounds. And this infection happens by, once it enters, it's going to multiply and then move into the xylem vesicles. And once this happens, it's going to create a blockage. And this bacterium also has virulence factors, which will suppress the plant's defense. And then we won't actually be able to see results whether the infection has occurred or not until the plants have finished growing. And I'll have to look at the flowering and the fruiting. On an internal level, what's going to happen is increased respiration will occur, and also a reduction of transpiration and photosynthesis. And then these pictures are just how the infection occurs. It's going to block the vesicles. And then this is also just my Bacillus subtilis. It's what it looks like. It's the gram-positive bacterium. So my predictions. My first prediction is that by inoculating the soil, the Bacillus subtilis will create a biofilm around the roots. And for this, I'll be testing about every 3 weeks just to make sure a biofilm is produced. And I'll be doing that by using the Congo red agar method. And once I actually have the biofilm produced, that's when I'm going to inoculate the soil with the Ralstonia solanacarium, which is the next prediction, which will, our solanacearium will try to infect the plant, but it's going to hit a blockade being the biofilm. And this, again, I won't be able to see until after the plants have grown and then observe them from there. The third prediction is that the tomato plant will not contract bacterial wilt disease and actual improved growth will be seen. And this, again, will be noticed after the plants have finished growing. And then, so my timeline, I'm going to be starting within the next few weeks. It gives me 4 months to allow my plants to grow, and hopefully I'll be able to observe them and notice improved growth rates. All of my supplies were met within the $150 budget. And in conclusion, what we're going to see is a relationship between the bacillus and the roots of the tomato plants forming a biofilm. And this will ultimately inhibit the bacterial wilt disease and positive plant growth will be seen. And then just some impacts on how this is affecting everybody, well, farmers, is that chemicals are really ineffective in preventing bacterial wilt disease, and if it does do anything, it doesn't permanently help the plants. And we're seeing that in the tropics, we're losing up to 70 to 100% of the crops. And this disease is actually moving into cooler temperate regions, which we can see from the maps. And if this study proves to be like a positive result, then we'll see that Bacillus subtilis will be a new safe and natural defense in preventing diseases in plants. And that's it. Thank you.